So during this session, we're basically going to go over mitosis, meiosis, and more stuff related to like cell cycle regulation, and even touch on like the bit of genetics that we've been doing with like the Punnett squares to kind of check for understanding on those sort of things. So first thing I would like to talk about then is the cell cycle. So can someone basically just give a brief description of what they think the cell cycle is? And you can use your mic or you can type something in the chat. Or if you don't know, just say don't know. Okay, and that's perfectly okay. So this diagram is basically an outline of the cell cycle, which is basically just the life cycle of a cell, right? So this is the cycle that that cell is going to continuously go through throughout its lifetime. So the first couple of steps that we talked about were G1, S, and G2. So do we know what the collective name for all of these stages is? And is it before mitosis or after mitosis?
Before mitosis?
What did you say? Sorry.
I think it's before mitosis.
Yes, you're absolutely correct. So these are happening before mitosis. And G1, S, and G2 are collectively called interphase. So all of these steps happen before we get to mitosis where we're actively dividing. Very good. And so now we're going to briefly go through G1, S, and G2 because it's very important that you know what's happening in each of these stages. So what happens in G1?
Forming in a chromatin form, and they're like.
Yeah, so you're absolutely right. So remember, before we get to mitosis in G1, S, and G2, we are in chromatin form. So that's our unraveled form of DNA. And in G1, we're normally functioning, so that goes along with the chromatin bit. So our DNA has to be unwound and accessible. And we're also growing, right? So we're growing to our normal adult size. Okay, so that's G1. So what happens in S phase then?
Duplicate.
Yeah, so this is DNA synthesis or DNA replication. So we double the amount of DNA, and that's going to be really important when we get to mitosis because when we, split, we need to have equal amounts of DNA in both of our cells. And then G2, what happens in G2? So in G2, again, we're growing more. We're getting, extra big because we're getting ready to divide. And we're also synthesizing those materials that are needed for cell division, like the microtubules, right? Those are really important. Okay, so G1, S, and G2, that's all interphase. It happens before mitosis. And then when we talk about cell cycle regulation, we talked about some proteins specifically that are involved in cell cycle regulation. Do you remember what those were called? I'll give you a hint. Cycle has a lot to do with it. Did you say cyclins?
Yes, I don't know if I said it right.
Okay, it cut out on me, but yes, correct. So the cyclins are what are ultimately responsible for cell cycle control. And there are 3 checkpoints, right? We talked about the G1S checkpoint. So like before we, replicate DNA, we need to make sure that nothing's wrong with the DNA. So that's the G1S checkpoint. And then there was a G2M checkpoint. So before, we're actively dividing, do we have all of the stuff needed to make 2 daughter cells? So that's where the G2M checkpoint comes in. And then we have a checkpoint here in between, metaphase and anaphase as well. So that's in your notes. Make sure to go over those details. But the cyclins are these proteins that are involved in cell cycle regulation, and they're produced along with the cell cycle. Okay, so then what happens when cell cycle control goes bad or goes wrong? And so we have these 2 genes that we talked about. We talked about proto-oncogenes and tumor suppressor genes. So can anyone tell me what proto-oncogenes are responsible for? Or tumor suppressor genes? What are their normal cellular roles? Okay, so a proto-oncogene is responsible for promoting cell division, so it kind of acts as like an activator for that. A tumor suppressor gene, on the other hand, inhibits cell division. So if we look at this picture right here, the proto-oncogene basically acts as the gas. It's like, yes, we got to divide, to do this. While the tumor suppressor gene acts as the brake and is like, well, hold up, we're not going to actively divide right now. So you can also think of it in terms of that. But sometimes we can break these genes and things can go very much wrong. So that's what happens with the oncogene. So when we break the proto-oncogene, which I think Dr. Arnett referred to this as like the proto-oncogene is the helicopter mom, it's regulating everything that, is going on in the cell, while the oncogene is the party mom. So the oncogene basically is a broken proto-oncogene, and it's a cancer-causing gene because it just allows for proliferation of the cell like uncontrollably. So it doesn't have that same regulation. And what collective disease does lack of cell cycle control cause? And it's in the picture, cancer, right? Cancer is basically just, uncontrolled cell cycle, uncontrolled cell division. So you just get a bunch of cells that have damaged DNA and, they just proliferate, and that causes a bunch of problems for the body, as you might imagine. So yeah, make sure to review these in your notes. That these are new concepts, but they still are important. So definitely take a look at those. Okay, vocabulary check. So here's something that's a little bit more familiar. So what's the difference between chromosomes and sister chromatids?
Isn't a chromosome 2 sister chromatids put together?
Yeah, so basically, and we see this in this picture, right? So when we go through S phase of interphase, we duplicate our chromosomes, and then we have this. We have the sister chromatids bound together by a centromere, and like you just said, that makes one chromosome. But when we have the unduplicated version, it's also called a chromosome. Very good. And so then what's the difference between chromatin and chromosomes?
Chromatin is like unraveled. Meanwhile, chromosomes are basically bunched. They're formed.
Yeah, so the chromatin, like we said earlier, was the unraveled form of the DNA, while the chromosomes is like the raveled, very put together form. And if you remember what Dr. Arnett said, it's like putting things into boxes and kind of organizing things better. So if that analogy helps you, also use that. So in which stages of the cell cycle do we see chromatin?
G1.
Good. Yeah, so G1, S, and G2, collectively known as interphase. Very good. And in which stages of the cell cycle do we see chromosomes?
Metaphase?
So, yes, you do see it in metaphase, but you also see it just at the beginning of mitosis. So, we are chromosomes starting in prophase, so at the very beginning of mitosis, even until the very end. So you could just say mitosis collectively for that. Because if we're talking about cell cycle stages, that would be like the end stage. So if we go back to our graphic, but good, yeah. So just keep that in mind. So then mitosis, so we'll briefly go over this. What is the purpose of mitosis?
Growth and repair.
Yes, very good. So mitosis is for the growth and repair. So like when we cut ourselves or we, scrape our knee, that's the process that's ultimately going on to, repair. And what are the stages? So the stages that you need to know, of course, are prophase, prometaphase, metaphase, anaphase, and then telophase. So that's why I typically remember it as PPMAT. So that helps me remember that. I hope that helps you. If it does, definitely use it. And so what I want to talk about basically is just kind of what goes on in each stage. So can someone describe to me what happens in prophase?
The chromosomes are condensed.
Yes, very good. So chromosomes condense. So now we've went from chromatin to chromosomes. So that's an important thing. There's also one other thing that happens during prophase.
Do the spindle fibers start forming in prophase?
Yes, very good. That's the other thing. So we have our centrosomes, and that's where the spindle fibers come from, and they're going to start to, migrate over, and as they do that, the spindle fibers form. Very good. So then what happens in prometaphase? Okay, so in prometaphase, then the nuclear envelope is going to disintegrate, or it's going to break down, and our chromosomes are going to attach to the spindle fibers. So that's what you see in this graphic right here. Okay, what happens in metaphase, which is our next stage?
Everything lines up in the middle.
Yes, very good. So our chromosomes are going to line up at the metaphase plate or metaphyseal. You'll hear it called quite a few things, but they just line up in the middle. And then anaphase.
The sister chromatids are pulled apart.
Yes, very good. So they're pulled apart to opposite ends of the cell, and that's what we see happening in the graphic. And then finally, telophase.
Cytokinesis.
Yes, good. So in telophase, we start to see the cleavage, and that's what you see right here, the division happening. And our nuclear envelope is going to, start to reform. And then eventually cytokinesis will happen where we have that division of cytoplasm, and then we form two identical daughter cells, which that's important to remember. That's the product of mitosis, identical cells. Which that's important to remember because when we get to meiosis, we see that that is definitely not the case. Very good. So again, remember PP-MAT. Okay, so meiosis then, let's talk about this one. So because this one's a little bit more complicated, but if you know the stages of mitosis, it definitely makes it easier to understand meiosis, especially meiosis 2, and we'll get to that. So what's the purpose of meiosis? Okay, so the purpose of meiosis is sexual reproduction, right? And generating that diversity. And that was definitely not the case with mitosis, which was just growth and repair of bodily cells, and we wanted everything to be the same. That's not the case with meiosis. We want things to be different. And what are gametes? Because we know that these are involved in meiosis. Okay, gametes are sex cells, right? Remember that the female generates the eggs and the male generates sperm. So those are the two types of sex cells or gametes. And are gametes haploid or diploid?
Haploid.
Yes, very good. And what does it mean to be haploid or diploid? What does that mean?
Only half of it is present.
Yeah. So like when we look at diploid organisms, they have, two copies of the chromosome, while haploid only has one copy of the chromosome. Good, or like one set. That's typically what you see, and that's why diploid is typically represented by 2N, and haploid is typically represented by N. So they have half. Yeah, good. Because when we take two haploid, cells, we take our sperm cell and our egg cell, and we combine them. What's that process called? Yeah, so that's fertilization, right? And that fertilized egg then is called a
Zygote.
Yes, very good. Good. Okay. So in class, we actually looked at how the formation of sperm versus the formation of eggs is different. So who can tell me how they're different? Or like what stood out to you in that process?
Sperm only has one X chromosome, and is that okay? And eggs have two X's.
Okay, so when we're talking about like female and male, so like males have, an X chromosome and a Y chromosome, and then females have two X's. But what I'm referring to here is like how with the sperm, so like, we undergo, meiosis and we're going to produce, 4 sperm at the end, and they're all going to be equal. Like when we form the eggs, something weird happens and we only end up with 1 at the very end. Do you all remember when we talked about how like when the division occurs, so like when we're doing cytokinesis, one egg cell gets like the lion's share of like the cytoplasm and the organelles, and that happens both times that division occurs, so we only end up with 1 egg at the very end, and like she's really big. Do you remember that?
Yes, I remember that now.
Okay, good. So that's what I'm referring to. And that's also important to note because, like you said, the X chromosome, that carries a lot of information, and the egg actually has, half the DNA, but it also has all of the organelles that are necessary for that cell to survive. And the sperm is really only bringing, the DNA because of how it looks. So remember when we saw a picture of, a sperm cell in class, it was basically just like a little piece of DNA attached to a tail. And that's not the case with the egg cell, definitely. So just keep that in mind. But very good. Okay, and then stages of meiosis. So this was actually our in-class activity. So you should have a really good study guide to use for the exam. And I sent out an email. I'm going to be developing kind of like an outline to help you all, guide your studying. But the in-class activity will be essential for studying meiosis, because if you can draw it and write like a short description of what's happening in each stage, then I would say you're pretty well prepared for the exam. So we can briefly go through some like major differences, but because we just went through mitosis and you seem like you all understand that really well, then this really shouldn't be a problem for you. So the only thing I really want to go over is meiosis 1, because in meiosis 1, something weird happens, especially with prophase 1. And can you all tell me what happens in prophase 1? There are certain types of chromosomes that like to pair up together and exchange information. Could somebody add a little bit more detail to that?
Is that called crossing over?
Yes, very good. So in prophase 1, our homologous pairs are formed, and crossing over is going to occur between those. And that's really important in generating the diversity, right? We see at the end, because they're going to exchange information when they pair up. Good. Okay. And then we see in metaphase 1, our homologous pairs are going to line up at the metaphase plate. And then in anaphase 1, what's happening? So our homologous pairs are going to be pulled apart, right? And then we go through telophase and cytokinesis, which kind of looks the same. So meiosis 1 really is where the big difference is between meiosis and mitosis as far as stages go. Because if you look at meiosis 2, it's really similar to the process of mitosis because you don't have the homologous pairs and whatnot. So definitely practice these. Go back and refer to your in-class activity. Make sure that you got all of your stages correct. And if you didn't, we posted a key in the eLearn page, so you can go look at that too and check. And if you have any questions, you can always let one of us know. Or if you'd like to go over it one-on-one, schedule a tutoring appointment or something of that nature too. There you go. Good job. So just keep that in mind and definitely look over these stages. Okay, so let's get to the newer stuff. So let's get to genetics. So a lot of terminology in this unit. So it's definitely important that we understand these terms and what they mean. So let's go through that really quick. So who can tell me what a gene is, just generally?
An inherited trait.
Good. Yeah. So like a gene is basically a piece of DNA that encodes for like a trait or something like that. And we know there are tons of traits like eye color, hair color, height, etc. Like the list goes on. So then what is an allele as it relates to a gene?
Is it like the coding? Oh, yeah, I don't know.
Okay, yeah, so like the differences, right? So an allele is like a variation in the coding of a gene. So where someone might have the allele for blue eyes, someone might have the allele for brown eyes and stuff like that. But yeah, you're right. Good. So that's what an allele is. And we saw how important like tracking alleles were with like Punnett squares, which we'll see one of those examples in a minute. Okay, let's look at heterozygous versus homozygous. So what is heterozygous? What does that mean to be heterozygous?
The alleles are different.
Yes. Oh, very good. So if you're heterozygous, you have different alleles. If you're homozygous, then what does that mean? Good, your alleles are the same. Good. So then dominant versus recessive. So what are the differences between those? So if I had a dominant allele versus a recessive allele.
Dominant is always expressed, and recessive is only expressed whenever it's with another recessive.
Very good. Yeah, that's a really good description. So remember, she said that dominant is like the bully allele. It has big or loud expression, while the recessive needs a buddy. Good. And then genotype versus phenotype. What are the differences between those? Okay, remember our genotype is the alleles that we have. So if we're, big B, little b, that's our genotype. And then, yes, phenotype is the physical expression or the characteristic. So if we're talking about eye color and, we look at the expression of, our big B, little b, then that would be, brown eyes in the example we did in class. Good. Okay. So it's really important to understand all of those terms because, they're just going to keep showing up and they're going to show up in your genetics packet problems. And so it's really important to know what they mean because if you're reading the question and you come across a term that asks you to do something specific for the question, it's just going to be a mess if you don't know what it means. And then it's going to be a mess on the exam if you don't know what it means. So terminology is definitely important here. So then we need to talk about like principle of segregation. We didn't talk about independent assortment yet, so don't worry about that. But can somebody just give me like a brief layout of what Mendel's principle of segregation is or what it states? And I know it's hard sometimes to put these things in your own words because it can be a little bit hard to understand it sometimes. But I included a picture right here that basically explains Mendel's principle of segregation. But remember, we talked about, yes, okay, so when we have the formation of gametes, those alleles, which are discrete units, are going to separate or segregate independently of one another. So, a gamete is going to only carry one specific characteristic if you also want to think of it that way. Very good. And that's what you see right here in this picture. So, we have, the meiosis 1 and the meiosis 2, and then we see the separation or the independent of alleles in each, gamete. Good. Okay, so let's look at Punnett squares, which are everybody's favorite. So this Punnett square has already been done, so we're going to kind of work backwards from how we did in class. So first, I'd like to ask you, what's the trait being investigated in a scenario? Like, what are we looking at?
The color of the flower.
Yes, very good. And from what you can tell about this Punnett square, what do you think is the dominant allele and the recessive allele?
The dominant would be the purple, and the recessive would be the white.
Very good. Yeah, because we see that wherever we have the big B, which is represented, by the purple, we have, the purple color. And then we have, wherever we have the little b in combination with another little b, we have the recessive phenotype shown. Very good. Okay, and the next thing I'd like to ask you, are the parents homozygous or heterozygous? I'm sorry, did someone try to speak? It cut you out a little bit.
I said heterozygous.
Yes, very good. So they're heterozygous because they have two different alleles. Very good. Okay, and then what we talked about today in class, I also included some of these. So what is the genotypic ratio and what is the phenotypic ratio? So in order to solve these problems, we first need to know, what does genotype mean or what does phenotype mean? See, this is where this really becomes important, like in terms of terminology. So we said genotype, was basically your alleles. So what's our genotypic ratio here? So how many different genotypes do we have?
3.
Good. We have 3, right? Because we have big B, we have big B, and then we have little b, little b. Yes, Madeline, so this would be a 1 to 2 to 1, right? And we would probably write, the type of genotype underneath the ratio as well. So like in this case, we would do big B, colon, big B, colon, little b. And that would be our 1 colon 2 colon 1 ratio. Does that make sense to everybody how we did that?
Yes.
Good. So then, what is the phenotypic ratio? So we said phenotype is physical expression. So like, what are the traits actually being expressed? So do we have the same ratio or is it different? Okay. So your genotypic ratio is not always the same as your phenotypic ratio, and this is a prime example of that. Because when we look at the genotypic ratio, it is 1 to 2 to 1. When we look at phenotype, so like purple flowers versus white flowers, we see that this is a 1 to 3, so 3 purple to 1 white. So this is a 3 to 1 ratio. So they're not always the same. So that is really important to note. Okay, is this an example of simple dominance? That's one of the inheritance patterns. So is this simple dominance or is this another type that we talked about called incomplete dominance?
It's simple.
Right, yeah, because we don't have an intermediate phenotype. It's like, if it has the dominant allele, it's going to have the dominant trait. If it has the recessive allele coupled with another one, it's going to be, the recessive. Good. Okay, and then what is the probability or the chance of producing a homozygous recessive flower?
25%.
Very good. Yeah, so our homozygous recessive, remember, is our little b. So we have, 4 out of 4 chances, we have 1 of those. So 1 divided by 4, and then multiply that by 100 to get the percent, it's 25%. Very good. Did a really nice job of working through that. So it's important to, practice these and remember to finish that one example that we ended on in class. So like the one about the the aliens having like gray eyes. It was an incomplete dominance example, so make sure you work through that. Okay, which brings me to this. So incomplete dominance is definitely a different inheritance pattern. So how does this differ from simple dominance in your own words? Like, how would you describe it?
There's two dominant traits and they're both being expressed.
Right. So how Dr. Arnett liked to put it, the people who were kind of, yelling across the chip bowl and it's just all kind of getting muddled. And so you get like this intermediate type of idea. And that's what happens basically with incomplete dominance. So like, both alleles are kind of, contributing almost equally and you kind of get this intermediate phenotype, which is what you see with the heterozygotes. So the ones that have the two different alleles from those two different traits. I have like the one we looked at in class and the one that you see right here is like with the red flower and the white flower. Well, if you have an individual or a flower that has, one red allele and one white allele, put those together and you get an intermediate phenotype, which is pink. So that's how it gets a little bit different. And we will definitely go over different inheritance patterns, and you'll see that it gets a lot more complicated than just simple dominance. But just continue working through those packets. And, you can always email me if you have any questions or problems with those. But does anyone have any questions about the stuff that we've talked about so far? Or do you feel pretty good about it? Like, how's that going? Because our exam is coming up really soon. And we definitely want to be cognizant of that. I believe it's actually, next week, next Friday. Feeling good, not so good. And I'm going to get a snapshot of attendance. Nervous. Yeah, so exam time always has me nervous too, especially in my own classes. But definitely, guys, just keep working at it. If scheduling tutoring appointments is what you do, definitely do that. There are a lot of biology tutors that can help you out. In addition to me and Jasmine, we are also resources for you to use. And I'm going to develop that study guide and have that sent out to you all. We will also upload a Kahoot soon, so you'll have that for practice. But the important thing is that you start studying now. So, go through your notes, identify your problem areas, and maybe stuff that you struggle with or that you haven't reviewed in a while. But definitely start going through that now so that you can spread out your studying over, quite a few days and, not wait until the last minute to study because cramming is never, and I mean never, a good idea. So are there any questions about the material that we've covered so far? I think you guys did a really good job, especially with mitosis and meiosis. Very pleased with that, so just keep up the good work. But if you don't have any questions, that's all I have for the session. Thank you all for coming, and I hope this, helped you. And if you ever need to review it, it will be on the YouTube channel, of course. If you'd like, I could even send out the PowerPoint. I know I put a lot of questions on here, and you can go back through the PowerPoint and kind of ask yourself the questions. Would that be something you're interested in?
Yes, please.
Okay, that sounds good. So I'll send it out right after I get off of here. Okay, well, have a great weekend, and don't forget to start studying and finish those problems that we talked about in class.